Good morning, students. Today we are going to discuss about the chapter 5, Principle of Inheritance, and here you see the inheritance of one gene. Inheritance of one gene can be explained by monohybrid cross. The cross between two parents differing in one pair of contrasting character is called monohybrid cross. That means when two hybrids, two pure line plants are crossed with one contrasting character, such as tall and dwarf. It is called monohybrid cross. Cross tall and dwarf pea plant collected seed and grew to generate first hybrid generation. So when tall and dwarf pea plant are crossed together, the first generation hybrid plant is called F1 generation or filial generation. F1 plants in monohybrid cross always tall and none were dwarf. For other trait also, F1 generation resemble only one parents and trait of other parents were not shown. So in F1 generation, all offsprings are tall, but dwarf plants are not found in F1 generation. Self-pollinated, when F1 offspring self-pollinated in F2 generation, and it produces tall and dwarf plant in the ratio dwarf, that is out of 4 plant 1 dwarf plant and out of 4 plant 3 tall plant. So F1 generation one parent trait shows, and in F2 generation both parent trait are shows in the ratio 3:1. So here we found that in F1 generation all plants are tall, but when F1 offspring are self-pollinated, in F2 generation they produces in the ratio 3:1. Here 3 are talls and 1 is dwarf. Next here you see, so Mendel proposed something that is stably being passed from parents to the offspring or to the next generation through gamete, and he denoted it as factor, and it is now known as genes. So genes and factors are unit of inheritance and contain the information required to express particular character or trait. Genes which code for a pair of contrasting traits are called alleles. Alphabetically, symbols were used to denote the characters, T, capital T for tall and small t for dwarf. Plan pairs of alleles for height. That means for a particular character such as tall, here two genes are found, they are called alleles. For tall, capital T both, for hybrid, capital T small t, and for short or dwarf, small t and small t. Mendel proposed true breeding tall or dwarf plant, identical or homozygous alleles pair of capital T or small t genotype. So Mendel by self-fertilization produces two types of pea plant on the basis of its height. If it is pure tall, then its gene composition will be capital T, and if it is dwarf or short, its genotype is always small t. So descriptive term used tall or dwarf as phenotype. And Mendel found phenotype of heterozygous are capital T small t of F1 was same as parent with capital T, and proposed in a pair of dissimilar factor, one dominant, the other, and hence called dominant. So when two plants, one tall and one dwarf, are crossed in F1 generation, which one is dominant? Capital T is dominant or tall is dominant, while in F1 generation, recessive traits are not expressed as character, short is not found in F1 generation, so it is called recessive trait. Next here you see Mendel's law of heredity. So actually there are 3 laws of inheritance, and all of them are called principle or laws of inheritance. First one is law of dominance, second one is law of segregation, and third one is law of independent assortment. So first one is law of dominance. According to this law, characters are controlled by a discrete unit that is called factor, and now it is called gene. Factor occurs in pair, and in a dissimilar pair of factor, one member of the pair dominant and the other is always recessive. That is used to explain the expression of one only of the parental character in monohybrid cross in F1 and expresses both in F2, that in the ratio 3:1. So law of dominance, we can say that every gene has two alleles that can code for a character or trait. Second, one allele is always dominant, means it will always show in F1 generation. One allele is recessive, means it will be masked by the presence of the dominant allele in F1 generation. So up to this, it is called law of dominance. Next, you see here when pure tall and pure dwarf, the genotypes are capital T, because in a somatic body it is always twice in chromosome numbers. Here two genes are given. Here also capital T. When during gamete formation it become haploid. So here capital T and small, here haploid gametes, that is small t. These both gamete, male and female gamete, are cross fertilized, and in next offspring or in F1 generation, it possesses the gene composition capital T small t. It is called hybrid, but the character evolved, that is always tall, not dwarf. But when the F1 hybrid capital T small t is self-fertilized, here in the F2 generation, it produces capital T, capital T, capital T, capital T small t, capital T small t, and small t small t. So here we see in case of law of dominance, up to this, this type of hybridization experiment is performed. Pure tall cross with dwarf, the F1 hybrid produces always tall. It is called law of dominance or dominant trait. Next one, we discuss about monohybrid cross. This cross is applicable in both law of dominance and law of segregation. In law of dominance, up to this, we describe it as a law of dominance. But in case of law of segregation, here, when the F1 hybrid is self-pollinated, here the gamete is found, capital T and small t, it form gamete, one capital T, one small t. Similarly, the female offspring also produces gamete, capital T, small t. When these two are crossed together, it produces tall. Similarly, it also produces tall. Similarly, here one t is come, capital T is bind with small t, it also produces tall. And last one, here capital small t and small t when crossed together, it produces dwarf. And the genotypic phenotypic ratio, monohybrid ratio is 3:1. And what is the genotypic ratio? That means gene composition. Here is 1:2:1. Next, this type of cross is called Punnett square. Here, production of gamete and formation of zygote can be given or described in the form of Punnett square. It is developed by British scientist Reginald C. Punnett, and graphical representation calculate probability of possible genotype in genetic cross. So in genetic cross, we describe the probabilities with the help of Punnett square. So this type of square is called Punnett square. Here gamete on two sides, top row and left row. So here the parent plant, from these gametes are formed. These gametes are formed, and these gametes are when in F1 generation, it produces capital T small t. In F2 generation, since we found only capital T small t in F1 generation, when this is done selfing, then it produces in the ratio in F2 generation 3:1. Next, here also, to define the homozygous or heterozygous condition, we can test with the help of test cross. What is test cross? When F1 hybrid is crossed with homozygous recessive parent, it is called trace cross. Here you see the capital P and capital P small p. It is the homozygous plant, and it is also homozygous plant. So here, either this alternative or this one alternative. It is homozygous recessive white. It is homozygous recessive white. So here you see this plant, whether it possesses capital P or capital P small p. Here, if we this one we select this genotype capital P and small p. Here is crossed with recessive parent, that is white pea flower. Then here it is the F1 hybrid and it is the recessive parent. Then its composition will be 50% are of capital P small p and 50% are white. Similarly, in case of cross with capital P with small p, that means when the dominant and recessive are crossed together, it produces in F1 generation, all of them are capital P small p. This F1 offspring, when crossed with recessive parent, it produces 50% recessive and 50% heterozygous as purple pea flower. So to determine their homozygous or heterozygous condition, we perform test cross. Next here, second law is called law of segregation. It shows that in monohybrid cross, we found that in F2 generation, we got 3:1 ratio in case of tall and dwarf plant, and 3 talls and 1 dwarf. Why it occurs? Because due to law of segregation. In law of segregation, chromosome pairs separate during meiosis, so that each gamete receive one allele for their given trait. Here you see the two chromosomes are different, two paternal, one paternal and one maternal chromosome. During meiosis, they exchange their segment. So during gamete formation, the alleles can be distributed in the gamete in equally. So here you see the 4 gametes are formed with different characteristics. So this is the law of segregation. So in F2 generation, we get at a shorter plant or dwarf plant in the ratio 3:1. So law of segregation states that when a pair of factor for a character brought together in a hybrid, they segregate or they separate during the formation of gamete. Why? Alleles do not blend or both characters recover. That means although in F1 generation, dominant character is expressed, but the recessive character does not blend with the dominant one. It gets expressed in F2 generation. So factors which is present in parent, they separate during gamete formation and receives only 1 or 2 of factors from those gametes. So homozygous parent and they produces one kind gamete, and heterozygous parent they produce 2 kinds of gamete, and each one have one allele with equal proportion. So here is the monohybrid cross, capital T small t plant crossed together in F1 generation. They are heterozygous because their gene composition are different. So when they are selfing in F2 generation, the gametes are segregated, they are separated. So this one is now, it is easy for this gamete to express itself. So it is expressed in this in dwarf form, and here the ratio will be 3 tall and 1 dwarf. It is the monohybrid ratio. 3:1 ratio is called monohybrid ratio. And here how many pairs of contrasting character were crossed? 1 pair of contrasting character, both height, but one tall and other dwarf. Up to this it is called law of dominance, and from this to this it is called law of segregation. So here I also give the monohybrid cross. Here again, when tall and dwarf are crossed together in F1 generation, capital T small t heterozygous conditions they are formed, and when they are self-fertilized, then they produces 3 tall and 1 dwarf in the ratio. The monohybrid phenotypic ratio is 3:1, and monohybrid genotypic ratio 1:2:1. Why it is different from this ratio? Because as here genotype is considered. Genotype means gene composition. 1 means capital T, only 1 is present, and capital T small t, 2 are present here. although they are phenotypically, all of them are tall. And here one represent the dwarf small t. So here today in this class, we have discussed Mendel's law of inheritance. There are three laws of inheritance: law of dominance and law of segregation. In both these laws, monohybrid cross is considered. That means one pair of contrasting characters are cross hybridized. Here in up to F1 generation, the monohybrid ratio is applicable. In F1 generation, only dominant trait is expressed, although the recessive trait is not completely blended or they are vanished. It is not like that. But in law of segregation, when this F1 hybrid self-fertilized, the gametes separated, so they are it is easy for them to visible. And so in law of segregation, here the ratio will be, monohybrid ratio will be 3:1. And in our next class, we are going to discuss about inheritance of two genes, because up to this we only discuss about inheritance of one gene. In law of dominance, we are going to discuss about the inheritance of two gene and dihybrid cross. Thank you, student, for attending the class.